Hello friends, welcome to another video. So today we will discuss about a very important topic in the pharmacology that is known as mechanism of action of insulin. So first of all we will discuss the mechanism of action. We must know what is insulin. Insulin is composed of what components? So first of all, insulin is a peptide chemical. So again, second question is coming in our mind. What is peptide? So peptide basically a kind of molecule that is a polymer of amino acids. So very huge number of amino acids that will combine and that will produce complex molecule that is known as peptide. So insulin is basically made up of peptide. Insulin is a peptide chemical. Insulin is secreted in our body by the beta cells in the islets of pancreas. Beta cell will secrete the insulin and then insulin releases on our body. When insulin released by the beta cell, that insulin taken up by our body cells, that will work in our body cell in such a way that it will decrease our body glucose level. So our main motto is how insulin will decrease our body glucose level. What the action that is produced by the insulin that the body glucose will decrease. So first of all, how insulin will synthesized in our body? Insulin is synthesized in the beta cells of the islets of pancreas as a pre-insulin from which 24 amino acids are removed and that will produce pro-insulin. Okay, pre-insulin में से 24 amino acid remove होने के बाद जो molecule बनता है उसको हम कहते हैं pro-insulin. The connecting peptide of the insulin, that is also known as C-peptide, that is split off in the Golgi apparatus, and the connecting peptide and insulin molecule both will stored in the granules of the cell. And when the insulin will become released in the blood, that C-peptide will also released together in that blood. Another fact we have to remember is the most homologous insulin to human insulin is pork insulin. So अब हम बात करते हैं हमारे मेन टॉपिक की, that is mechanism of action of insulin. So first of all, इंसुलिन का जो रिसेप्टर होता है वो अल्फा और बीटा कॉम्प्लेक्स से बना हुआ होता है. 2 alpha and 2 beta complex that will combine and that will form the receptor of the insulin. In alpha complex, that is the binder and that is the extracellular component of the receptor. Beta complex is a transmembrane component of the receptor. In alpha complex, the insulin will bind. This is an insulin receptor, this is alpha complex. Insulin will bind on the alpha complex and that insulin will produce a response in our cell. Beta complex is a complex that is associated with tyrosine protein kinase and which is a phosphorylated complex. So when insulin will attach on the receptor, that tyrosine protein kinase, that will phosphorylate IRS protein. The IRS stand for insulin receptor substrate protein. That IRS protein will become phosphorylated. So that IRS protein will become phosphorylated, that is an phosphorylated IRS protein. That phosphorylated IRS protein that will produce two actions. The first action is stimulation of secondary messenger. The secondary messenger like phosphatidyl inositol triphosphate. This phosphatidyl inositol triphosphate that will produce one function that is known as activation or inhibition of the enzymes in the inner side of the cell. And the second action that is produced by phosphorylated IRS protein is activation of phosphorylation cascade. The phosphorylation cascade, when become activated, that phosphorylation cascade that also produce activation or inhibition of enzymes, that also activate GLUT, that is a glucose transporter. This activated glucose transporter will goes into the cell membrane, and in this cell membrane, the activated GLUT that will produce a kind of channel that will permeable for glucose. Through this channel, glucose will enter into the cell, and extracellular glucose, the amount of extracellular glucose will become decreased by this action. The phosphorylation cascade again do third action, that is known as activation of MAP kinase. MAP stand for mitogen activated protein. Mitogen activated protein kinase become activated by phosphorylation cascade, and mitogen activated protein kinase that will activate DNA, and DNA, after the process of transcription, it will converted into mRNA, and mRNA, after the process of translation, it will converted into protein. Protein that do the functions in the cell like cell proliferation and cell differentiation. So these two functions that is produced by this protein. Okay. And the second thing is that when the insulin act on this cell, okay, the receptor become inactivated after the action of insulin. That inactivated receptor undergoes into the process of internalization. So the receptor will translocate into the inner side of the cell, and that receptor again goes into the two actions. That is known as, first action is known as recycling of the receptor. That again receptor become recycled, and again it is ready for taken up of insulin. And the second process that the receptor goes is degradation. That the receptor will become degraded. So this is all about the mechanism of action of insulin. This is a very, very important mechanism that we have to remember. So this is all about the mechanism of action. I have briefly covered the topic of mechanism of action of insulin. So आपको ये वीडियो अच्छी लगी हो तो इस वीडियो को लाइक कीजिए, इस वीडियो को शेयर कीजिए, और अगली वीडियो सबसे पहले आप तक पहुंचे इसके लिए इस चैनल को सब्सक्राइब कीजिए, कमेंट कीजिए। तब तक के लिए बहुत बहुत धन्यवाद।